This is what an amino acid looks like. You don't have to draw it out, okay? I'm not going to ask you. It's not a chemistry course. But I just want you to appreciate a little bit that we have a carbon, we call it an alpha carbon, and it has 4 bonds to it, as it's supposed to. We'll also have a carboxylic acid group. So there's going to be a carbon here, and still there's a double bond, but 1, 2, 3, 4. There's always going to be 4 bonds there. There's an amino group where we're going to have a nitrogen there with a couple of hydrogens. And every amino acid that we have will have this whole setup. They are identical. The only thing different is going to be this R or radical group, and that could change. It could be a ring here, it could be just an H here, it could have a COOH, it could be a lot of different things. And that's what makes one amino acid different from the others. There's 20 different amino acids in our body. All are identical except for that R group. All are used to make proteins. They're letters in the alphabet, they're going to make words. If you have a long word, if you have a long chain of amino acids, we call it a polymer. We have essential amino acids. I abbreviate amino acids AA. It's not Alcohol Anonymous. Okay. But amino acids, there's essential ones. Essential means that our bodies, there's certain ones that our bodies can make, and there's certain ones we cannot get. We have to get it from our diet. So we have 9 essential amino acids that have to come from our diet. We can't make them. Then we have non-essential amino acids, and there's 11 of those, okay, that our bodies can make. So if you look at this, the yellow of all these different, and I'm not going to ask you to draw them out, all right? This is not the course. This is, in fact, I don't even think they want you to do that in regular chemistry. You like this kind of stuff, go into biochemistry. You'll memorize all this stuff, and you'll have wonderful fun with that. Not my forte, okay? It's just the concept I want you to understand. You don't even have to know about the names of them. I mean, just recognize that, you know, if I say 3 amino acids, glycine, tyrosine, and cysteine, do this. Well, you know what I mean? At least you know what amino acid is. And they have their own abbreviations. The yellow is what's the same on each one. It's kind of a reverse picture of what you saw before. But the blue is the radical, the R group, and it's different for each amino acid. Like I said, it could be very short, it could have a couple rings to it, but that's what makes one amino acid different than the other. We have 26 letters in the alphabet, we have 20 amino acids. There's 88 keys to a piano. But think about the 88 keys to a piano, how many songs you could come up with, how many revamping of songs you could come up with. With the alphabet, think of just 26 letters, how many words you could come up with in books and novels and stuff. Same way here. Amino acids is only 20, but you could make a lot of different things with those 20 amino acids. These are the different amino acids. Again, you won't have to memorize it, but just to show you at FYI, here's the essential ones and here's the non-essential ones. There are 2 essential ones that children also have to have that we don't need it as adults. We end up making it later on because our livers get made and they get more efficient. So that's what it's just showing you. There's a couple over here that children also need too. So they need something more in their diet. But again, I'm not going to ask you to memorize that. It's just an FYI. This is the part we didn't get a chance to talk about yesterday, but we will in next Friday. This is a ribosome. This is your kitchen, and we're going to make the brownie. Here's the mRNA. This is a tRNA, transfer RNA. Here's mom that's bringing the eggs, the amino acid that's over here. It's going to go into the ribosome. It's going to add this to the bowl of whatever ingredients are already in there, and you're going to produce this long chain of amino acids, your brownie. Meanwhile, mom attaches over here and comes off with amino acid gone. She's not she's leaving without eggs because it was put in the bowl. Okay? So that's what it's showing you over here, is that the building blocks, which are amino acids, are going to make proteins. This picture here is showing you translation, and that's what I was hoping to get to yesterday, but we'll definitely do it next Friday in class. Okay? A peptide is any molecule that's composed of 2 or more amino acids. So this is a peptide. It's 2 or more amino acids. A peptide bond is the bond that actually is going to bound the amino acids. So going back to here, that's a peptide bond. The white there, peptide bond, peptide bond, peptide bond. The nomenclature, pretty easy to understand. If there's 2 amino acids together, we call it a dipeptide. Tripeptide is 3. If you have 4 to 15, a small amount of it means oligo. Oligopeptide is a small amount, 4 to 15. A polypeptide is up to 50, and then we call it a protein once it goes over 50. So it's just a nomenclature. It's pretty easy to understand that, okay? What can proteins do? We've seen some of it already. It can make structure. When you get into skin, it's going to make keratin, which is going to make sure that your skin doesn't get dried out so much. Collagen is going to be a protein that's going to be part of our connective tissue that's going to give you the tissue its bulk. Okay? Communication. There's going to be hormones that are made of proteins. There's going to be receptors that are on the cell membrane. They're all proteins. So that something binds to it. It's the keyhole of the key, right? It's going to develop the channels in the membrane, the cell membranes, so that sodium, potassium can pass through those tunnels, those channels. All enzymes are made of proteins. It'll also make antibodies. Antibodies are going to help with the immune system, so it'll help fight infections. It'll also help you to clot blood. It'll also deal with movement, all right? The ability to, a cell can change shape. Something like a white blood cell can turn into like an amoeba and change shape to squeeze on through in between the cells of blood vessels to get out to where the infection is. And we also could use proteins to help glue cells together. Those things we're talking about with tight junctions and desmosomes and stuff. All right? So what I'm trying to show you is proteins is what you are. If you don't have proteins, you're just dead. You can't survive. You need proteins. So that's why we're going through the whole process of how transcription, translation occurs. Animals need proteins. A lot of bacteria needs proteins. So it's nice to know how bacteria works with transcription and translation, because some of those bacteria you don't want on your body, and a lot of the antibiotics are going to be able to target certain parts of where proteins are made in a bacteria so the bacteria die. So this stuff, transcription and translation, is going to come back to haunt you when you deal with microbiology. You need to understand the concept of this because we need to understand that. Now, proteins are structurally organized in these ways: primary, secondary, tertiary, quaternary. So basically what it is, the primary is going to be all the amino acids tied together. Okay, and it's a long chain. This could be alanine, and this could be cysteine, this could be another cysteine, this could be tyranine, this could be serine, this could be lysine, leucine, this could be, methionine, this could be another cysteine, this could be alanine, this could be phenolphthalein, and so forth. That's our primary structure. So think of it as like a long line, okay, in order. Then the secondary structure is we're going to do this. We could turn it into something else. Maybe it wants to go into like a spiral. So take like a slinky and stretch it all out as one wire. Then you want to coil it up. That's going to be our secondary. Then you could actually take the slinky and you could bend it like this. That's our tertiary structure. What's going to hold it to stay like this? All hydrogen bonds. Hydrogen bonds are going to keep it in that way. Why does it do that? Because, you know, in a certain way, alanine needs to bind over to here. It can't happen, so we've got to bend it, and maybe now the alanine can now touch the methionine over here. It creates it so that it's able to function properly. Then we take this chain, and we could have a chain over here, a chain over here, another one over here, and another one over here. And I put 4 there. You don't need to have 4. You could have 3 chains glued together, 2 chains. But when you do this, this is now called the quaternary structure. So you got primary, secondary, tertiary, quaternary. And if I wanted to, I could put an iron element right here in each one of these chains. And what I created was basically a molecule of hemoglobin. One oxygen attaches to here, one oxygen attaches to this iron, one oxygen here, one oxygen here. And that's in red blood cells, it's loaded with hemoglobin. So it's just showing you primary structure, tertiary structure, primary structure, secondary structure, tertiary, they're trying to show you that there's a iron in there, in this particular case. And then you're going to have, let's say, the hemoglobin, which is a quaternary structure. Okay? So amino acids could be used more than once to make different proteins. Like letters in the alphabet. We're using the same letters, we could reuse them. The precise sequence has to be there, otherwise it's a totally different thing. A good example of this is we use 3 letters here. What does that say? Cat. Would be nice if an A and P was as easy as that. Okay? It has to, for me to get that image of a cat in your head, I have to spell it this way. If I change it and just change one letter, you're not going to see a cat in your head. Even if I change it and reverse things, you're going to see something totally different in your head. So it has to be precise. If the reverse, it'll be a totally different structure. If there's one that gets replaced, it's a totally different structure. Totally. I could have, let's say, a 20-letter word, and just one letter will change the whole thing. So it has to be precise. Insulin is 51 amino acids long. Okay? It's a protein hormone, and every amino acid has to be that order. Otherwise, you're not going to make insulin. It's something else. And in most cases, it's probably garbage. Or it'll be a deformed insulin structure that just won't function. All right? Like 88 keys on the keyboard. You know, if one key's out of tune, it just doesn't sound right. This is a red blood cell loaded with hemoglobin. someone's got sickle cell anemia. Now, hemoglobin is this structure here. 4 different chains. Each chain is about 145 amino acids long, making this structure almost 600 amino acids long. 600 amino acids. Sickle cell is that one amino acid out of that nearly 600 has been replaced by a different amino acid. And instead of making this kind of shape, it makes this by 1. All right? In this case, glutamic acid is replaced with valine in the 6th position. Just that 1. So that's why it has to be precise. I mean, like I said, if I showed you the cat and hat, 1 letter makes a big difference. Okay? The shape of the protein greatly affects the ability to recognize and bind to other things. All right? If you denature, if you destroy that protein because it's in a hostile environment, it gets destroyed. You're not going to work. It's not going to work properly. This is why when you have a stomach virus and you have a fever, your body temperature goes up to over 100.4. The higher it goes, it's going to change the proteins in your body. Take, for instance, an egg. You crack an egg, you put on the frying pan, okay? The white of the egg is something called albumin, protein. It's all protein. If you put that on a skillet that's burning, it changes. It goes from a liquid to a solid. Look what happens even with Jell-O. It's all collagen in there. When you change the temperature, it changes. So if those are proteins, the proteins in your body are going to work the same way. That's why you feel so awful when you get sick. Because your body just can't work as fast as it did before. So you want to keep that down. That's why our biggest concern, when they get like 104 temperature, we got, 104, 105, when you have heat stroke and stuff, we got to cool your body down. And that's the reason why. Same thing with pH. If the pH goes too high or too low, it'll also destroy these enzymes. Not just enzymes, all proteins. Okay? Questions so far about this? Sugars. These are rings. All right? They're all made from 6 parts carbon. 6 parts carbon, 12 parts hydrogen, 6 parts oxygen. If they have one ring, and they're trying to show you carbons here, carbons here, carbons here. I kind of showed a little bit when we were talking about rings and the 5' and 3' when it deals with DNA, and we're going to do DNA replication. Again, I'm not going to ask you structures and draw these out, but they are rings. Okay? If they're single rings like this, we call them monosaccharides. These are the smallest things that can actually get absorbed into our bloodstream from our diet. Glucose, galactose, and fructose are examples of that. You should know those. This is what they look like in rings, but a lot of times when they show it, they like to just break it, and then you can see it like this. So it's not in a ring form. But why I put this up here is to prove to you about these carbons. They have 4 bonds each. And because we're attaching them to hydrogens, that's what makes it an organic structure. All right? You won't have to draw these out. Don't worry about that. But just understand that they're organic. We also have it where there could be 2 rings tied together. In this case, it's a disaccharide. And I showed you this earlier with sucrose. It'll break up into glucose and fructose. This one they're trying to show you is an example of lactose, milk sugar. And when we break up this, you're going to have 1 molecule of galactose and 1 molecule of glucose. We can't absorb that in our bloodstream. It's just too big. So we have to have an enzyme that's going to break that. If this is lactose, guess what enzyme is going to break that? Lactase.
Excuse me. So this is made up of two monosaccharides.
Correct. 2 monosaccharides will turn into a disaccharide. All right? And this is lactose. All right? Just give it, you won't have to draw them out, but I'm just saying that lactose, you should know that lactose and the one I showed you before is sucrose, those were disaccharides. All right? Mastering this stuff now will make your life easier in AP2 with digestive system, because we'll be talking about all these enzymes, breaking them up and what they do and how they do it. So again, we talked about dehydration versus hydrolysis. It's a similar thing over here, which I don't want to go over again, but it's the same thing we talked about, catabolism, anabolism. Okay? So these are the 3 important disaccharides you should know. Sucrose is table sugar, lactose is sugar in milk, milk sugar, and maltose is grain products, wheat, that kind of stuff. And this is what they're made up of. Okay? When they're monosaccharides, we definitely can absorb them. Polysaccharides. These are long chains of saccharides, or sugars, okay? Potatoes is a good example of what that is, okay? Potatoes are sugars. They don't taste sweet because the sweetness comes from the size of them. If they are just one ring monosaccharides, they're very sweet. Disaccharides, they're sweet too. Polysaccharides are long chains that kind of look like this. They're not sweet. Bite into a potato, tell me how sweet it is. Okay? It's just that we can't break that down as well in the mouth. We have to have other things later on in the digestive system to break it down. We can break it down. There's a sugar called amylase, but it's not as efficient as pancreatic amylase. So that's what happens with that.
Where did you make mention of maltose?
Maltose is here, which these are grains. So glucose and glucose make up maltose. All right, wheats, rice, that kind of stuff. All right, so they're in long chains. So we can have oligosaccharides. These are short chains of 3 or more, and polysaccharides, which are at least 50 monosaccharides long. Right? Again, they're pretty easy. Oligo meaning a short amount. Just nomenclature, semantics kind of thing. But in your readings, you might, why are they talking about oligo? Here I'm defining them all for you. Now, like I said, sweetness. They have to be broken down into the smallest rings, the one, monosaccharides for you to get the sweetness going on. So here's the important polysaccharides. Glycogen, starch, and cellulose. Glycogen is what's in our bodies. It's mostly in the liver, some muscle. So we have long chains, and like I showed you before, it's like an iceberg of sugar. And it's in our liver and some muscle. It's a storage site. In plants, we can either have starch or cellulose. Starch is digestible by us humans. Things like potatoes and rice and wheat. Cellulose is indigestible, but it's still in our diet. These are the things like roughage and fiber. It won't get absorbed. We can't use it for building things, but it's still important to make sure that our colon is actually strong enough to push things out. It's kind of like training ourselves, but making it real roughage so you can push things out and have a like prunes, that kind of thing. In prunes, there is sugar in there also, but there's also fiber. Okay? So this is just a summary, just so you can visualize what the difference between monosaccharide, disaccharide, and polysaccharide. Okay? Lipids. How we doing with stuff? Not too bad with this? All right? Okay. Lipids or fats.
A quick digestion question. So in like wheat and stuff, is there cellulose and starch